Okay, let's just dive right into this. Welcome to today's explainer. We're tackling something that honestly gives a lot of biochemistry students night sweats: cellular metabolism. Specifically, how our cells literally generate their own internal energy currency through a process called glycolysis. I'm your host, and I'm joined by my awesome co-host who's going to be stepping into the shoes of, well, you, the curious student.
That's right. I'm here to ask all those burning questions you're probably already thinking. And yeah, I gotta admit, looking at all those chemical pathways can be pretty intimidating.
Oh, totally. But I promise you, by the end of this explainer, you're going to see it as this brilliantly logical, step-by-step system.
So, here is our roadmap for today. We'll start with the whole concept of energy currency, then we'll break glycolysis down into phase 1, the energy investment, followed by splitting the sugar, and then phase 2, which is the energy payoff. Finally, we'll tie it all back to the human body and wrap up with a quick recap.
Whew, thank goodness for the recap. Memorizing 10 biochemical steps definitely feels like a lot to take in at once.
We've got you covered. We'll make it so logical you won't even need to rote memorize it. Let's get started.
All right, section 1, the energy currency hook, funding the cell. You know, whenever I look at cellular metabolism, my brain just sees this massive tangled web of carbon rings. It's so overwhelming. At the most basic level, how do cells actually stay alive and fund their daily work?
Think of glycolysis like a simple business startup. You know the old saying, you gotta spend money to make money. Whether you're flexing your muscles at the gym or firing neurons to study for an exam, your cells are doing work. And that work requires a very specific type of cellular funding.
And that funding is ATP, adenosine triphosphate. This is literally the ultimate currency the cellular business wants to stockpile.
So when we talk about energy in biological terms, we're really just talking about hoarding this ATP molecule?
Exactly. ATP holds free energy right there in its chemical bonds. When the cell needs to get something done, it just snaps a phosphate off that ATP to release the energy. The entire goal of the glycolytic pathway we're unpacking today is to manufacture this exact ATP currency.
Which brings us nicely to phase 1, energy investment.
Okay, wait, investment? If our whole goal here is to make ATP, why on earth are we investing it?
Because just like opening a real-world storefront, you need startup capital. Our starting material here is a 6-carbon sugar molecule called glucose.
Right, I see that. And right off the bat in step 1, an ATP swoops in and drops something off.
Spot on. That ATP attaches a phosphate group right onto the glucose. We are officially spending our energetic money.
But I have to ask the classic beginner question here. Why? Why are we using ATP right out of the gate? It just seems completely counterproductive to burn through your profits before you even start making them. It's such a good question. The answer is all about trapping the glucose. You see, when glucose enters the cell, it could easily just drift right back out. But by spending an ATP to slap this bulky, negatively charged phosphate group onto it, the cell effectively locks the doors. That phosphorylated glucose can no longer cross the cell membrane. It's trapped inside, completely committed to our business venture.
What's really fascinating is the specific machinery, the regulatory enzymes used to do this trapping. In most of your body's tissues, an enzyme called Hexokinase handles this job. It has a super high affinity for glucose, meaning it works hard even when glucose levels are pretty low.
But there's another one mentioned here too. Glucokinase?
Yep. Think of glucokinase as hexokinase's liver-specific cousin. It has a low affinity for glucose, so it really only kicks into high gear when blood sugar is surging, like right after you eat a big meal. It helps your liver soak up all that extra glucose to store it for later.
So let's quickly summarize the first 3 steps of our investment phase. Step 1, trap the glucose. Enzymes called kinases act like molecular postmen delivering phosphate groups, and hexokinase delivers that very first one.
Then step 2 is all about rearranging.
Right. An enzyme called an isomerase acts like a molecular architect. It takes that trapped molecule and subtly reshapes it from a glucose ring into a fructose ring, all without changing the actual chemical formula.
And then step 3 is commit. Let me guess, another kinase?
You got it, phosphofructokinase, or PFK. It delivers a second phosphate, which means we're spending our second ATP. This is absolutely crucial. It's the ultimate point of no return for the molecule.
All right, moving into section 3, splitting the sugar, diversifying our portfolio.
So we're taking our single 6-carbon asset and splitting it into 2 independent 3-carbon operations? How does a sugar molecule just snap in half? Enter our next enzyme, Aldolase. Think of Aldolase as a pair of molecular scissors. In step 4, it snips our doubly phosphorylated 6-carbon sugar right down the middle, leaving us with 2 3-carbon pieces. One is called DHAP, and the other is GAP.
But they're different molecules. I thought we wanted a streamlined, identical process. We do, which is exactly why step 5 brings back our molecular architect, an isomerase called triose phosphate isomerase. It rapidly converts the DHAP into a second molecule of GAP. So moving forward from this exact point, we have two completely identical 3-carbon molecules.
And that kicks off phase 2, the energy payoff.
Finally, it's time to see some actual return on this ATP investment. Let's cash out. Let's see how this all builds up. Now notice a really crucial detail here. From this point forward, everything happens twice. Ah, I see that. Because we split our 6-carbon sugar into those 2 3-carbon molecules earlier, right? Exactly. Every enzyme, every reaction, and every single payout from here on out gets doubled. We are officially tracking 2 3-carbon molecules, our GAPE, simultaneously.
Okay, looking at the very first step of this payoff phase, I'm seeing something new popping up. An NAD+ turns into an NADH. Where do the hydrogen atoms actually coming from? To make that happen? And honestly, what's the point?
That is an excellent observation. That magic happens in step 6. Let's break down exactly what's going on with those hydrogens.
The enzyme GAPDH basically strips hydrogen and high-energy electrons off our sugar and hands them over to a carrier molecule called NAD+, reducing it into NADH. And NADH is incredibly valuable. It's kind of like a metabolic check we can cash later for a ton of ATP.
Nice. And then step 7 is where we actually see the very first ATP produced?
Yes. Phosphoglycokinase performs what's called substrate-level phosphorylation. It grabs a phosphate off our sugar and slaps it directly onto an ADP, creating our very first ATP. And remember, this happens twice. Then, a mutase relocates the remaining phosphate, and an enzyme called Aldolase removes a molecule of water to create a really unstable, high-energy compound.
Which perfectly sets up step 10, the grand finale payoff.
Right. Pyruvate kinase cashes out that last phosphate, forming our final ATP and leaving us with our ultimate end product, pyruvate.
Let's bring this into the real world with section 5, the human body connection. The bottom line. Okay, let's zoom out a bit and do the macro accounting. We invested 2 ATP right in the beginning. In the payoff phase, we produced 2 ATP in step 7 and another 2 ATP in step 10, for a total of 4. So 4 minus 2. Our net ATP yield is exactly 2. Perfect math! The business is officially profitable. We have a solid net gain of 2 usable ATP per molecule of glucose.
But that's not all. The cell also nets 2 high-energy electron-carrying molecules of NADH, which we generated back in step 6.
Right, those were the checks we can cash later?
Exactly. If the cell has oxygen, it will literally take those NADH molecules over to the mitochondria, drop them into the electron transport chain, and generate a massive amount of additional ATP.
And finally, looking at the physical carbon product remaining from our original glucose molecule, we are left with 2 molecules of the 3-carbon compound called pyruvate. Correct. This 6-carbon glucose has been fully cleaved, processed, and transformed into 2 3-carbon pyruvates.
So here is how all of this biochemistry directly applies to human anatomy. Some cells in your body, specifically your erythrocytes, your red blood cells, completely lack mitochondria.
Wait, if they don't have mitochondria, they literally can't cash those NADH checks or process that pyruvate any further, right?
Precisely. Without mitochondria to perform oxidative phosphorylation, human red blood cells rely 100% entirely on this exact glycolytic pathway for their energy production. Without glycolysis, your red blood cells just couldn't survive to carry oxygen to the rest of your body. It's that important.
All right, section 6, quick recap for exams. Let's bring it all home with a rapid-fire review.
Okay, here's the bottom line ledger. For inputs, we start with 1 6-carbon glucose. We invest 2 ATP, we use 2 NAD+ molecules to pick up electrons, and we need 4 ADP molecules waiting around to be phosphorylated. For outputs, we end up with 2 3-carbon pyruvates. We generate 4 ATP, giving us that net gain of 2. We yield 2 NADH carriers, and we release 2 molecules of water as a byproduct of the Aldolase step. You nailed it! You have successfully mastered the inputs and outputs of the glycolytic economy.
But before we completely wrap up this explainer, I want to leave you with one final provocative question. We know what happens if the cell has oxygen and mitochondria. The pyruvate continues on to the citric acid cycle. But what if the cell lacks oxygen, or, like our red blood cells, lacks mitochondria altogether?
Oh, right. What actually is the next metabolic venture for that pyruvate when the standard path is completely blocked?
That is exactly what you'll need to explore as we dive into anaerobic metabolism and fermentation. Until then, keep analyzing, keep asking those great questions, and we'll catch you next time.